Okay folks, so as you can see here, we have the setup for doing the Organic Chemistry 1 lab at home for the liquid extraction experiment. Alright, so out of this little blue baggie that was inside of your kit, yes, I have blue gloves, I'm special, I have my own nitrile gloves. But you should be wearing, of course, whatever gloves came with your kit, or alternatively, whatever gloves you may have at home that fit better. Alright, so in this particular procedure, you are going to end up mixing blue dye with in water with mineral oil that has yellow dye in it, and we are going to do the extraction in this bigger jar. We've got little disposable Pasteur pipettes here, and we have our graduated cylinder for doing measurements. And lastly, in the event, as I was talking about last time that we had lab, in the event that your mineral dye here, mineral water dye, did not have very much, if any, food coloring in the bottom of it. See, this one has a lot of spotted coloring in it, but if we look at it more from a side view, we see that it's not very well dispersed. Okay, then what we will end up doing is actually going and making sure we do disperse it properly. Okay, so there is a very specific procedure for doing so, and so we're going to see if I can manage to get that done with you here. So now the challenge is opening this with one hand. Okay, no, seriously, I'm gonna see if I can somehow prop this phone up in a manner that will allow me to keep filming. Keep filming what I'm doing while you can watch. Whoa, don't fall over. All right, so hopefully, maybe can do that. Let's see here. That should probably work. Okay, so we open up this mineral with yellow dye. I closed it pretty tight the last time I played around with it. So if you don't have very much yellow speckling in the bottom, you should take your food coloring, open that package, pull out the yellow, open that, and put one drop of food coloring into the mineral oil. Now, before I go and start playing with the mineral oil, I'm going to follow the instructions from the procedure. I'm going to open my jar, put this over here for a second, and I'm going to measure out my 40 milliliters of my blue dye. And it says about 40 milliliters, so if it's not exactly 40 milliliters, that will be okay. For the purposes of what we are doing, I'm going to go with exactly 40 milliliters. And in order to do so, I'm going to use one of these pipettes and just bring this up to a meniscus at exactly 40 milliliters. Just like this. And there we go. Done. Okay. So, close that, set it aside, and pour that in. Okay, now, because I've already used one thing in here, I'm going to actually take this out of here for a moment. Yeah, that comes apart in case you didn't know. I'm going to rinse this with water. So that I make sure that's nice and cleaned out from the previous dye color. Now I'm taking a paper towel and I'm just rolling it up like that so that I can then twist it and briefly dry this out a little bit and briefly dry the outside. You want to be a little bit careful with these plastic pieces because they're plastic and therefore a bit resilient they are not necessarily so resilient that they will not break. All right, so I'm going to shove that aside for a second. Now the best way to mix this is actually, as much as I wanted to just do my own thing before, the best way to mix this apparently is this shearing method of sucking up the dye and then expelling it back out. So that's what I'm trying to do here. So we suck up the dye particles and then expel them back out with this micro pipetter to try to get it as all mixed in as we can. It's a bit arduous, but seems to be, from what we were told in the experiment, the best way to go about doing this. So, also stir this as I'm doing it. Alright, so we're going to keep going with this shearing thing. Try and wipe the dye off the sides a bit. Seems to have wanted to stick to the plastic a little. So dispersing the dye seems to be certainly a little bit problematic, at least. Which is unfortunate, but it's what we have to deal with. Alright, so. Alright, well, that's about as good as we're going to get at the moment. It's a little bit better dispersed. So we're going to see what happens with what we've got going on in there. Alright, so I'm going to also put the lid on it here. Give it a good shake as well. Honestly, I think shaking does a better job. Okay, let's open it quick. Do a quick pour. Alright, and the directions say okay, volume of all the yellow dye with the mineral oil, and so we end up recording that. I know how much it is, and now pour all of it in on top of the blue dye. Alright, so there you have it. Now that's on top of the blue dye. This is the lid for this chamber. I'm going to put this one back on top of here, and I need to get out another glass or jar of some sort to hold on to whatever happens. Alright, so this is going to be my receiver glass for what happens here. Alright, so if we look at this, alright, we see that we have two different layers, as we should. And actually, the layers are already kind of mixing a little bit. I don't know if you can tell or not with the light, but there's some mixing that's going on. So we have to take this and do 1, 2, 3, 4, 5 shakes. Kind of let it sit, let it separate, open it up in case there were any gas bubbles. And this is what you would normally do with something called a separatory funnel. So I'll have to make sure that I show you a picture of a separatory funnel at some point. Alright, but as you see, we kind of like swirl it a little, sort of try to induce a little bit more separating power. Alright, the layers are starting to separate a bit. Okay, and we're going to repeat shaking another 5 times. 1, 2, 3, 4, 5. And we're going to let it sit. And so upon standing, everything should separate. Now, we know in theory that oil and water should always end up separating from each other. We actually see it happen all the time when we do things like, I don't know, make a salad dressing, perhaps. You know, if we end up using something like oil and vinegar to make a salad dressing with spices or anything like that. So this is something that we've seen, and we know that can happen. In this particular instance, what we're actually seeing is, again, oil and water separating, but we're also seeing that if we have oil and water separating, we are also watching how a material that was mixed into the oil as best as we could might actually be perhaps even more soluble in the water layer. Because you see how there's a color change in the water layer. All right, so now what I'm going to do is I'm going to take this and, following directions, we are going to remove the one layer, the mineral oil layer, back into the mineral oil bottle using the pipette, which is what we are instructed to do. And so we have to do that pretty carefully. These pipette bulbs apparently only hold, I'm going to say, a couple of milliliters, so this is going to be a bit painstaking to do, but it'll get done. If you happen to have something that's maybe a little bigger that you could use to do this sort of process, you are welcome to do so, but just make sure that it's clean of any other contaminants, and that you know, that you're able to do this effectively. Definitely don't use a straw and try and suck it up. You don't want this in your mouth. It'll taste horrible. Just for any of those that are trying to be a little too creative. All right, and you can see that I've actually accidentally also sucked in some of the water layer, so I'm going to try and get that back out and put that back in here. All right, so, and sometimes if you're looking at it, if you look at it exactly at eye level, you're able to better pull up only the oil layer, or not. It really depends on how good you are at doing this. But you really want to try and be as precise as you can be. Now, the advantage of doing something like this with the actual lab equipment, such as a separatory funnel, is that a separatory funnel has in the bottom of it a little stopcock that you can open and close, just like on the bottom of a titration burette. And that means that if you wanted to, you would be able to instead drain the water layer out of the bottom instead of trying to pull the oil layer off the top. And that is oftentimes so much easier to do. Okay, so this is essentially what you got to work at doing here. Pulling that off of there. And it seems to take a while. Okay. All right, so that about does it. I am going to pour everything into my other glass so that now I've got my aqueous layer that is all green here in the other glass. Next, I want to measure out the rest of my blue dye. which is again about another 40 milliliters. I'm going to pour that back into my chamber, and I'm going to pour now my oil back on top of that and see if I do extract any more color. And repeat. All right, so you basically just go and you repeat this process one more time. Again, open in case there was any gas build up in there. All right, same thing, open again in case there's any gas buildup. Now, here's something else that can sometimes happen. Every so often, something like this, when you're trying to do it, it doesn't want to actually start separating. Now here we're actually starting to get some separation again, but every so often something like this says, nope, I don't really want to separate. I want to stay in emulsion. All right, so we can actually start seeing the distinct colors again here. So it's getting there, all right? And our expectation is that it will separate and that we will end up recombining once again our aqueous layers in our extra glass on the side. But one of the tricks that you can use to force separation of an emulsion that could form is actually to take either a saturated, well, it's actually best to do that, to take something like a saturated salt solution, and you can add, you know, like a milliliter or two, as long as it's going to be compatible with what's in your aqueous layer, some saturated salt solution, and actually toss that into your mix of your emulsion that could have formed. All right, so what's in a saturated salt solution? Well, you've got certainly aqueous component, water, and then you have ions, right? You have sodium ions and chloride ions, and it's saturated, so it's as much as possible that can fit into solution. Okay, so thinking about the properties of oil being nonpolar and water being extremely polar, and reasons for why an emulsion might form. Okay, so just to give you some extra hints with that, emulsions sometimes form because, well, with an emulsion, you know, you may have some compounds that are in the material that have properties that are similar to both oil and water, right? So you might have some parts let's say, for instance, with amino acids that are polar, that want to interact with the water, or that are nonpolar, that want to interact with the oil, or certain fats, or steroid compounds and such. Something like salt will force a process called salting out to occur. All right, so give that some thought, and you can probably come up with the reason why adding saturated salt solution will help in getting rid of an unwanted emulsion that could happen in a separatory funnel and a liquid extraction process. Okay, so that's the demonstration that I have for you regarding this whole liquid extraction. So this is everything that I wanted to show you at this point. You will have top layer, bottom layer to work through once again, and after that I would say that you are just going to need to, you know, submit the results that are requested on this particular experiment sheet, all right, the 3-page experiment sheet. And I'm also going to submit to you a couple of guidance questions to help you with understanding the process of what's going on when you're doing one of these separations, and some other problems that are going to just give you a little bit more thought-provoking questions in understanding what happens during extraction and washing techniques. Other than that, we'll see you soon.